Hello everyone. Good morning students. How are you? Okay, this is another Saturday and my name is Ram. Welcome to Easyhai exams, okay, where we'll be studying and solving the previous year NEET level questions, and we'll also be studying each of their options and making you clear about all the concepts. Okay, that's what we're going to do here today. My name is Ram and yeah, let's continue from where we left off last week. Okay. We did 10 questions last week. So here we go. This is where we start. Okay. So the very first question for today is from the previous year question paper of NEET 2021. Which of the following algae contains mannitol as a reserve food material? Okay. Now, before we start answering this question, I want you all to understand that we are all carbon-based life forms. Okay, we are all carbon-based life forms. That is, our entire body is basically made of carbon structures. Okay. Now, how do plants fix this carbon? How do they increase their own biomass? Through the C3 cycle. Yes or no? Yeah? Through the C3 cycle. They take in carbon dioxide and they fix it into their own bodies via the C3 cycle. They form glucose, and glucose goes ahead and forms what? Starch, and that's their food. And when you eat potatoes, when you eat any of the vegetables, you're eating that carbon source that the plants had stored for themselves. Okay, now this is what is happening. Now plants store it as starch. Okay, that is your basic angiosperms. They are storing it as starch. And then there are other types of plants also that come under kingdom Plantae, right? Kingdom Plantae, of which algae is also one of them. And algae is of three types. We have chlorophyceae or chlorophyta. Okay. We'll just clean up the workspace. You have rhodophyta, and you have phaeophyta. Okay, chlorophyta, which has green pigments, rhodophyta, which has red pigments, and phaeophyta, which has brown pigments. They're also called brown algae because of the pigmentation. Brown algae, red algae, and green algae. Okay, now they are asking mannitol. Mannitol is the reserve food material. Okay, mannitol is the reserve food material. Just for extra knowledge, mannitol is a sugar alcohol. Okay, it's basically a sugar that has OH groups attached to it. And since they have many OH groups attached to it, they are also called polyols. Polyols. Ols representing the alcohol group and poly meaning many. Okay. So that's about mannitol. Now they're asking which of these stores its reserve food as mannitol. Now in the NCERT textbook, it's directly given that chlorophyta stores its reserve food as starch. Okay, and rhodophyta, that is the red algae, stores its food as Floridian starch. And finally, phaeophyta. Phaeophyta, which is the brown algae, stores them as mannitol or laminarin. Laminarin. Okay. So we have to find out which of these. Is it Ectocarpus? Is it Gracilaria? Is it Volvox? Is it Eulothrix? Which of these four is a phaeophyte? That is, which of these four is a brown algae? That is what we have to find. That is something that you will have to remember. So we know Eulothrix and Volvox are chlorophyta. Okay. We know Gracilaria is a rhodophyta. And finally, Ectocarpus is a phaeophyta. That is, it's a brown algae. And since it's a brown algae, it stores its reserve food as mannitol. Okay, so what's the correct answer here? Ectocarpus. Ectocarpus is your correct answer. So there you go. We've solved one question, and we've also learned about the entire concept of how different algae stores its reserve food. We've also learned about a little bit about mannitol, that it's a sugar alcohol. There's no point going much deeper into it because NEET is not going to question you on the structure of mannitol and all. Okay, okay. I guess I can give you one more small bit of information. Mannitol is also used as artificial sweeteners. Okay, for people with diabetes, since this has low glycemic index, it's not taken up by the small intestine. Okay, so it does not increase the sugar level in your blood. So people with diabetes, okay, if you want to have chai, if you want to have tea, okay, now you cannot have sugar, so they use mannitol as a substitute, as an artificial sweetener. Okay, a little bit of extra knowledge there. All right, let's move on. Next question. What happened? All right, here we are. Okay, in spite of second question. Okay, in spite of interspecific competition, that is competition between two species. I'll explain on that. I'll elaborate on that. Okay, in spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? Okay, so what does the question say? The question refers to a scenario. Suppose there's a common area and there is a species X and there is a species Y. Okay, species Y, and both of them are competing for one particular resource, let's say R. Okay, now species X also needs R and species Y also needs R, so they're competing for each other. Like for example, when you guys are, okay, very good example actually, when you guys are competing for your medical seats, there are medical seat is the resource here and there are lots of people competing for it, so there is interspecific competition. Okay, interspecific, if you break down the word, inter is between and specific refers to species. So when the competition is between two different species, we call it interspecific competition. What are they competing for? For resource. The resource could be a habitat, a place to live, maybe a place to lay their eggs or raise their young ones, or food. Okay, or a watering hole. Watering hole is basically a small place where there's water and animals go to drink. Okay, it could be anything. People can, not only people, animals, organisms can compete over anything in order to sustain themselves. Okay, so that's where interspecific competition is. We've understood that concept. So the question asks, when there is such a competition between two different species, when there is such a competition, what is the mechanism they might have evolved? कि कौन सा mechanism evolve कर रहा है उन दोनों के बीच में, so that both of them might survive. Okay, now that is the question that they are asking. Now we've understood the question. Let's try answering them. Let's understand each of the, let's understand each of the options. Let's start with predation. What is predation? There's a tiger. The tiger predates upon, a deer. It's eating it, it's hunting it, it's eating it. That is called predation. Simple as that. Okay, what is mutualism? Now mutualism is a relationship between two species where both the species are benefited. Okay, if I have to give you very famous examples, there are lots of examples. Okay, you might have seen the birds sitting on top of rhinoceros. Okay, you might have seen the birds sitting on top of bulls, cows and buffaloes. Yeah, what's happening here? The relationship there is that there's this cow. Okay, let's say there's this cow. Okay, throw in some horns also. Okay, so there's this cow and there's a bird. Okay, there's a bird. And what happens is, obviously, there'll be lots of mites, there'll be lots of insects on top of the skin, and the bull cannot reach it. What does it do? the bird actually eats these insects. Okay, so it's getting its food, but at the same time all the infection or the itches or the scratches or any sort of rashes that might develop because of these bugs is prevented for the bull. Similarly, another example that I can give you is that of the yucca or yucca moth. Okay, now there's a specific plant, there's a specific cactus whose whose flower is actually pollinated by the yucca moth or the yucca moth. Okay, now what does the moth get in return for this? What happens is these moths actually lay their eggs inside the plant, inside the flowers. Okay, and when these moths, the larvae come out, okay, when they come out, they actually go up and they get nutrition from these flowers. Okay, the flower is giving oviposition. What is the resource that the flower is giving to the yucca moth? Oviposition. Oviposition. What is oviposition? Ovi meaning egg, position mean place. So it's giving a place for the yucca moth to lay its eggs. Okay, so both of them are benefited. The plant is being benefited in the fact that it is getting, it's getting pollinated, and the yucca moth is actually having the benefit that it has a place for oviposition, and also the larvae have something to eat when they hatch. Okay, so that is mutualism, a relationship where the two interacting species, both of them are benefited. Now that's mutualism. Let's understand what is competitive release. This might be a new term, Okay, competitive release is when, suppose again, this also comes in competition. Suppose there are two competing species A and B, okay, and what happens is, it has been seen that when you experimentally remove a species, okay, before we jump into that, I guess I have to explain a little more. Let's make the diagram a little more interactive so that you can imagine what's actually happening. So there's a species A, and and this is the entire area where it's able to live in. It's the entire area where it's utilizing its resource. Okay, and it's able to utilize only this much part. It's only able to utilize only this much part of the habitat that it's living in. And then there is a species B which is able to live in this entire place. Okay, since they are both competing, and B is a better competitor, it's able to compete for the same resource or for the same habitat better than species A. So it's able to exude, it's able to show its influence on a much larger area, Okay, but now it has been seen, there was research that was done. Okay, and in that it was found out when you experimentally remove this species, gone. Okay, so the influence that it exudes, the influence that the older species exudes is also gone. Okay, so what is happening? This species is competitively released. It's gotten a relief from the competition that the older species actually used to have. Okay, and now since the old species that was competing against species A is not there anymore, what happens? Previously, when the influence of A was only this much, it now extends all over this place because there's nobody to check it. So basically what's happening, A is being released from the competition of B. Okay, an example was the research work was done on barnacles actually. And it was seen that one species, it's not really important, I'm just giving you a very brief overview. Barnacles, there were two species of barnacles. One was seen only in the above the tide area, whereas the intertidal area was completely dominated by another species. When the species in the intertidal area was removed, it was seen that the one that lived above the tidal area started growing into the intertidal area also. Okay, so that is the example. This is doing it. Okay, competitive release. Now, resource partitioning. Now, you guys might have already understood now, since I saved this for the last, this is probably the right answer. And you guys are correct. It is resource partitioning, which is the right answer. That is when there is a high amount of interspecific competition. Okay, in order for both of them to survive, the mechanism that is evolved is resource partitioning. And it also makes sense. The name also makes sense. That is, the resource has been partitioned. Okay, they share the resource in such a way that both of them can co-evolve. Both of them can survive. Okay, both of them can survive. So let's understand resource partitioning as well. I'll give you an example, a very famous example from the islands of Puerto Rico or Puerto Rico. Okay, Puerto Rico. Now, what happens is, in the island of Puerto Rico, there are two different species of anole lizards. Anole lizards, basically lizards. Okay. What happened is, these two different species were competing for habitat. Okay, as I said, competition can be for many things. It can be for food, it can be for, a place to lay eggs, oviposition, it can be for food, it can be for watering holes. So these two different species of lizards were actually competing for a place for their habitat, where they would live, where they would forage, where they would find their food. Okay, so what happened is, over time it was noticed that one species of anole stuck to the trees, bushes, branches, and all those things. So if this is the land, okay, and these are the trees, okay, these are the trees, okay, this is the canopy. So what happened is, one species started inhabiting this part, okay, the upper area. Let's say they kind of became arboreal. I'm using the term very lightly. Okay, okay. So one species started inhabiting this part, the trees, the bushes, all the higher parts, and the lower understory, okay, the ground part, was inhabited by another species, species 2. Okay, so the anole lizards, which were competing for habitat, a place where they would stay, where they would live, where they would breed, where they would find food. Okay, since both of them lived in the same place, they were coming in competition with each other. They were coming in competition with each other. So what did they do? One species went up the trees and they lived there, and the other species lived on the ground and they foraged there. Now that is resource partitioning. That is, this entire habitat was divided up between two species, species 1 and 2, so that they both can survive. So we've understood the idea of resource partitioning. There you go. So does this make sense now? Interspecific competition, yes, both the species, anole species, were competing against each other. What is the mechanism that the competing species evolved? What did they evolve? They evolved resource partitioning for their survival, because both of them are now surviving in the same habitat. Okay, they do not come in the face of each other. So let's move on to the next question. Okay, genera like Selaginella and Salvinia produce two kinds of spores. Such plants are known as. Now, right off the bat, I'll tell you that Selaginella, Salvinia, they all come under your Pteridophyta. Excuse my handwriting a bit. Pteridophyta. Okay, they come under kingdom Plantae, and under that they come under Pteridophyta. Now, they're asking two different kinds of spores. Now, even if you don't know the entire concept, just looking at the options that we've given here, okay, we can directly answer this question. It's a very simple question. As I said in my previous lecture, I was talking about monoecious. Okay, I was talking about dioecious. Remember? And in that, I also spoke about heterothalous, right? Now, if you guys have actually heard that lecture, okay, if you guys have heard that lecture, you guys should be able to answer this question. Okay, without actually even knowing the entire concept, you guys can actually answer this question. Look at the question. Produce two different kinds of spores. So logically, what should be the answer? When I said different, hetero means different, okay? And spores, sporus. So what's the answer? Heterosporous. Let's go one step ahead. Homosporous. What did I say when I spoke about the same question? Okay, I also spoke about homothallus. Homothallus. What did I say homo was? Homo meaning same. Homo meaning same. So what is homosporous? These are plants that give out the same kind of spores. The same kind of spores. Understood? There you go. You've already answered the question. But then again, at Easyhai, we do not just leave our students with small tips and tricks. We make sure that their concept is also clear. So let's understand the concept also here. Okay. So what happens is, there is a sporophyte. And the sporophyte, what is the ploidy of sporophyte? Can you guys tell me what's the ploidy of sporophyte? Come on, write it in the chat. Is it 2n or is it n? Go ahead. Okay. You guys are funny. Okay, so cool. So yeah, where was I? Sporophyte. Okay, so we have a sporophyte. The sporophyte is, as a few of you have said, it is diploid. It's 2n. Okay, it's diploid in nature. Diploid. Di meaning 2, ploidy meaning ploid meaning ploidy. That is how many sets of the same chromosomes are there. So they are diploid, 2n. Now what happens is, this sporophyte produces a spore. Okay, and in homospory or homosporous, what happens is, this spore. Okay, first of all, how is this spore produced? It's produced by reductional division. Reductional division, also known as meiosis. Okay, if it's undergone meiosis, now tell me this spore that is produced, what is its ploidy? What will be its ploidy? Go ahead, tell me. Ploidy is, yes, of course, it's n. Very good, very good, very good. Okay, now what happens is, this spore goes ahead and produces a body that has both the antheridium, the male part, and the archegonium. Archegonium, that is the female part. Now this condition, okay, they further go on, they produce gametes and then they fuse and then they give rise to a sporophyte. These two are called gametophytes because they produce gametes. Simple. Gametophyte. Okay, now since they produce the same kind of spores, this is called homospory. Now imagine, instead of it happening like this sporophyte, this meiosis. Okay, wow, I cannot recognize my own handwriting. Okay, anyways, so the spores produced are of two kinds. Okay, one is the male spore and the other is the female spore, and they both go ahead and give separate bodies. One becomes the antheridium, the other becomes the archegonium. Okay, the other, let me write it properly. The other becomes the archegonium, the female part. Okay, this is female, this is the male part, and they both give rise to bodies, and they both give rise to separate bodies, and then they both form gametes. So you see, the difference happens here. The spores produced, there are two kinds of spores that are produced. Okay, one that develops into the antheridium and the other that develops into the archegonium. Now that is called, my dear friends, what is that? Yeah, yes, it's heterospory. Yes, heterosporous. So we've understood the trick how to make out questions without actually knowing the entire concept, and we've also understood the concept behind it. Okay, that's the best part about having a teacher. You know, they tell you all the connected parts. When I was studying in college, what happens was our teacher used to ask us to create mind maps. Okay, wherein you could connect information, and you know, just by learning one question, you're learning two or three different other concepts. So in a way, you know, it's thanks to my teachers that I've pretty much adopted their own teaching style. So yeah, that's a little bit of history. Okay, let's go on. Okay. The site of perception of light in plants during photoperiodism is photoperiodism. What is photoperiodism? Okay. Now, you guys, I'm in Delhi right now. Okay, and the summers are pretty harsh. I'm pretty sure it's same everywhere across India right now. And the summers, as you might have noticed, in summers we have longer days. The sun sets here around 6:37. Okay, even around 7, we have a very good sunlight outside. In fact, there were times when I took my afternoon nap and I wake up and I see that there's light outside. So I'm like, okay, suppose if I slept at 2, I'll be like, okay, there's light outside, so probably it's 4. Turns out I've been napping for 5 hours. It was a full-fledged sleep. Yeah, those days have also happened. So, but at the same time, what happens in winter is about, by the time it's 5 o'clock, it's pitch dark. Okay, so you see the length of the day has changed. Okay, with seasons, sometimes within days itself, the length of the day has changed. Okay, and that is called photo period. Photo period. Photo meaning light, period meaning time. Okay, simple as that. Break down each of the words so that you can understand the meaning without actually, even if you didn't know it, you can actually make out the meaning by breaking down the words. Photo meaning light, period meaning time, duration. Okay, now that is photo period. Now what is photoperiodism? Okay, just like how I was reacting to the light outside, seeing that there is still light outside, so probably it is probably it's just 4 or 5, and I went back to sleep. Just like that, plants also I know it's a weird example, but yeah, plants also respond to this light periods. Plants also respond to the light periods. How? Now in angiosperms, what happens is the flowering phenomenon in angiosperms is dependent on the amount of light that is there. Okay, and based on that, we have 3 types that are given. That is long day plants, then we have short day plants, and we also have day neutral plants. I think the naming, the terminology is pretty self-explanatory. Long day plants, meaning they require a lot of sunlight in order to start flowering. Short day plants, they require very less period. Excuse me. Yeah, they require a very less period of sunlight in order to flower. And day neutral is, it's okay, no matter how long we'll flower. So that's the sort of idea here. Okay, now how do plants sense this photo period? How do plants sense these changes in photo period? Who is responsible? Now this, please, my dear friends, please note it down. It's called phytochrome. This is extremely important. Okay, phytochrome, also called cryptochrome. These are photosensitive molecules that are found in the leaf. So the site of perception of light, where is light perceived? In the leaves, because it is in the leaves that we have the phytochromes. Okay, and that's why our answer becomes the leaf. Okay, so you've understood what photoperiod is, you've understood what photoperiodism is, you've understood what's long day plants, short day plants, day neutral plants, and you've the most importantly, you've understood phytochrome and cryptochrome. These are, let me just write it down so that you guys can note it down. Okay, these are photosensitive. There is a very long process which happens, that when light hits them, they change near infrared, far infrared. There's lots of things, and then they move to the nucleus. So lots of things actually happens. So you don't have to know that, you just have to know that these phytochrome and cryptochrome are the molecules that are responsible, and they are found in the leaves, and that's why the site of perception of light becomes the leaf. Okay, and that's how you answer this question. Let's move on to the next question. Okay. Okay. Plants follow different pathways in response to the environment or phases of life to form different kinds of structures. This ability is called elasticity, flexibility, plasticity, or maturity. Let's break each one down. Okay. Elasticity, I'm pretty sure everybody has taken a rubber band in their hand and yanked it so hard that it hit themselves and then scream out in pain. Elasticity, something that comes back to its original position. Okay, a rubber band like this, you stretch it like this, it comes back to its original position. That's called elasticity. Now, what's flexibility? Now, there's a structure. Okay, there is a straight structure. Okay, how much can it bend from its original position? Okay, without breaking. That's called flexibility. Okay, an iron is not that flexible. A rod of iron is not really flexible. Okay, it takes a lot of effort in order to bend it up. Okay, but at the same time, gold, it's highly malleable. Okay, you can bend it up and it's not going to come back, but it's not going to break either. Okay, but at the same time, you think of something very brittle like a rod of plastic. If you try to bend it, it snaps, it breaks. Okay, now that's flexibility. Now plasticity is the answer. Okay, what happens is plants are sessile. What is sessile again? That is, they are rooted to a particular substratum. They do not move around. They are rooted to a substratum. Soil, rooted, soil is the substratum, the ground is the substratum, and they grow. Okay, now since they cannot move about, what happens is when the environment changes, they also have to respond to that change in environment. Okay, now suppose you're sitting in a room, okay, and suddenly the room starts getting really hot, okay, and you do not have an AC, you do not have a fan. What do? You just get up and leave. Okay, you leave to a colder area. That's what we are seeing right now. Most of the people are going to Himachal and Uttarakhand because it's getting really hot here. Okay, it's getting really hot in the plains, so people are moving to the mountains, having a good vacation, posting on Instagram, and coming back. Similarly, but plants do not have that luxury. Plants do not have that luxury of, when the environment changes, they cannot just get up and leave. Okay, I do not like this environment anymore. I am going to the hills. The hills are calling me. The mountains are calling me. They cannot do that. So what happens is there are certain phenotypic changes that occur. Phenotypic changes. Okay, phenotypic changes that are shown in plants. And this phenomenon wherein they change their phenotype in order to adjust with the environment, or in order to respond to the changes that have happened in the environment, and so that they can survive better in that changed environment, is called plasticity. And hence our answer for this question becomes plasticity. That's your correct answer. And before we move on to the next question, let's also discuss the last this thing. It's called maturity. Maturity is, I have lots of jokes right now, but I'm not gonna crack them. Maturity is, okay, in biology, strictly in biology, what happens is in maturity, you have two phases in the life cycle. You have two phases. You have juvenile phase. It's a U, okay? Juvenile phase. You have a reproductive phase. Reproductive phase. When your organism goes from juvenile phase is when, as the name suggests, child phase, it's not reproductively active yet. Okay, when it moves from juvenile to reproductive phase, that is, this individual can start reproducing. That is called maturation. That is called maturation. Okay, and at this phase, it's called maturity. Okay. On Instagram, we have lots of this posts that come, maturity is when you realize so and so, so and so, so and so. Okay, so yeah, that's not the maturity that we are talking about here in biology. In biology, it is when an individual starts becoming sexually reproductive and reproductively active. That is called maturity. Okay, let's move on. Okay, it's match the following now. Match the following. We have Christy thylakoid, centromere, and cistern. Now, my dear friends, the ones who tuned in last week on Saturday, the ones who tuned in might already know what's a centromere, because we spoke about it at length. You know, when we were talking about metacentric, submetacentric, acrocentric, telocentric. So that's actually an advantage for them, because you've already seen you already know what centromere is. Okay, so what is centromere, my friends who actually attended the previous class? Okay, it's the primary constriction in chromosomes. For my friends who are joining in new, for my dear students who are joining in new, I will obviously explain it again. Okay, you have a chromosome. Okay, now what are each of these arms called? They are called chromatids. I'll show it to you. Let's take a different color. Let's take a red color. Okay, now if I were to take just this part, this one single arm, let's call it arm, okay, that is called a chromatid. Now, since these two chromatids come from the same chromosome, these two are called sister chromatids. Okay, because they both come from the same chromosome. Okay, and these two sister chromatids are held together by a primary constriction. Okay, they are both held together by a primary constriction, and that primary constriction is called your centromere. Okay, it's called your centromere. Now, a little bit of extra information which I also gave in the previous class is, on top of the centromere there is a button-like structure. Called the, should I take a different color? Okay, this blue thing that I'm drawing, okay, that's your button-shaped structure called your kinetochore. Okay, so while meiosis happens or mitosis happens, basically cell division happens, what happens is the chromatids have to be separated. Yeah, the chromatids have to be separated. Now, for that separation to happen, spindle fibers come in. Okay, spindle fibers come and attach themselves to the centromere via the kinetochore. Okay, so while learning centromere, we are also learning about kinetochore. Oh, gone. We're learning about kinetochore. Okay, now kinetochore is a button-like structure that is present on top of the centromere, and they help attach the spindle fibers to the centromere and help in the separation of the sister chromatids while cell division is going to take place. Okay, so we've understood what centromere is. What is centromere? Centromere is the primary constriction in chromosomes. Now, in Match the Following, I'd like to tell my friends a small little, a small trick, a cheat of sorts, wherein once you've already identified a matching pair, try to find if, try to eliminate other options. So that you can save time in your exams, which can be important. Time is obviously the most essential commodity when you're going for a competitive exam. And what happens is, if rather than sit and decipher each of these and then try finding out all of these, okay, what you can do is, once you've matched one pair, okay, you've matched C with 1, okay, try to find out which of these options have C and 1 in the same thing. More often than not, it happens that you've already cracked it. Let's see if we've cracked it just by knowing centromere is a primary constriction. So we have to see which one has C1. Okay, there you go, we've actually cracked it. It's this. So even without actually knowing what is cristae, what is thylakoid, what is cistern, we've already answered the question. There you go. Okay, but again, we're not going to leave here with half knowledge. We need the full knowledge, we need the full concept. So I'll be teaching you the entire thing. Okay, what is cristae? Now, I'm going to say a very famous line. Mitochondria is the, please type it down. Mitochondria is the dash of the cell. What is it? Mitochondria is the, thank you. Thank you. It is the powerhouse of the cell. Yeah. Mitochondria is the powerhouse of the cell. This is a very famous line which everybody says. In fact, lots of memes have also been made on this. Okay, yeah. So yeah, in our powerhouse of the cell, what happens is it's a double membrane structure, right? Now the outer membrane is flat, it's disc shaped, okay, but the inner membrane has folds like this. Okay, the inner membrane has foldings like this, infoldings. Now these infoldings are called your cristae. Okay, so what is cristae? They are the infoldings in mitochondria. Okay, so we've cleared that. We've also understood what's cristae. Let's see if our technique actually worked. If this is in fact the correct answer, A should be paired with 3. Yes, it's correct. A is paired with 3. Correct. Okay. Now what's thylakoids? Thylakoids. Okay. From mitochondria, we are coming to chloroplast. Okay. Chloroplast is again a double membrane structure, right? Chloroplast. Let's use green color here. Okay. Because chloroplast is associated with chlorophyll, and chlorophyll is green colored pigment. But I hope this is actually visible. You know what? I'll stick to black. Okay. Okay, it's a double membrane structure, and inside the lumen of the chloroplast, you have disc-shaped, flattened disc-shaped structures called the thylakoid. Okay, it is these thylakoids that are disc-shaped, flattened membranous sacs in the stroma of the plastids. Okay, flattened membranous sacs in the stroma of plastids. What's plastid? Now, as I said, this is the chloroplast. Now the chloroplast has fluid-like structure, that a colorless fluid. The colorless fluid is called stroma. Okay, and this each of these stacks, you see, not stacks, each of these sac, each of these membranous sacs, flattened membranous sacs, each of these are called thylakoids. But what happens when you look through the microscope? It is that you find them as stacks, not sacs, stacks. So basically stacks of sacs. Okay, these stacks are called your grana. Okay, the singular is granum. Okay, that's the granum, and these stacks are actually connected by a lamellae-like structure. They are called stroma lamellae. Stroma lamellae. Okay, so we've actually labeled and identified each of the structures that are in the, what is this entire structure? It's called the chloroplast. Okay, that's the cell organelle that we are studying. Okay, finally, cisternae. What is cisternae? Okay, let's also check this option. Thylakoid is B to 4. Yep, B to 4, it's correct. So, and by default, this becomes, cisternae becomes the disc-shaped sacs and Golgi apparatus. Okay, I'll just draw and show it to you what they mean. Okay, the Golgi apparatus, we draw it like this. Okay, this is how we draw it. They are again membrane bound. They are cell organelles. Okay, and cell organelles are membrane bound. Okay, and each of these sac-shaped structures, okay, is called your cisternae. And all these things together is what we call the Golgi complex or the Golgi body or complex. All right. So I hope everything is clear. I hope it's clear what is cristae. I hope it's clear what's thylakoid, centromere, cisternae. I hope all of these are clear. And also the small trick that I told you, once you clear once you clear, one option and you're very sure, yeah, that option is correct, try to match that option and try to eliminate other options. Okay, that will save you very precious time in competitive exams, because you have chemistry and physics as well, and you are going to need, when there is numericals, okay, you are going to need time. You are going to need time, and wherever you can shave off a few precious seconds, do it. Okay, uh, you know once you've matched it, instead of breaking your head over, okay, I don't know what's sister name, I don't know what's the entire matching, try to eliminate options, and one more often than not, usually one is enough, more often than not. I'm not saying that's the case all the time. No, I'm not going to say it. Okay, it might have been possible that C1 was there for more than one option. Okay, in that case you would have to look for other options as well, you'd have to solve other matches as well. Okay, but wherever chance is possible, try to shave off time, try to save time and use it elsewhere. Okay, and there's a certain level of psychological impact as well when you save time, when you know that you have time in the bank, when you know that you have buffer time. It keeps you calm and it helps you think and also remember and recall better. Okay, I think that is probably the biggest tip that I can give you, is to keep yourself psychologically sane, keep yourself calm, because in any exam, when you start getting stressed, you start losing it, and even the questions that even the answers or the information that you actually remember, that you've actually learned, it becomes difficult to recall. We all, I myself, having given the exam, I know how it goes, and I have all India rank 1 in BHU's entrance exam. So yeah, having sit here, I know how it goes. So just a few tips and tricks being passed on. That's it. All right, let's move on to the next question. Okay. Again, it's another match. Okay, we have cohesion, adhesion, surface tension, and guttation. Okay, let's understand each of these. Okay, and by understanding each of these, uh, you know, let's say processes, we'll try to answer and match. Okay, let's start with, let's say, where shall we start? Okay, let's start with guttation. Okay, let's start with guttation. What is guttation? Okay, guttation is associated with plants. Okay, what happens is, during the nights, we all know that the stomata is closed. Okay, the stomata closes down, and since the stomata closes down, transpiration does not occur. So the excess water that is there in the plant is not given out. Okay, it is through transpiration that water vapor is also given out by the plants. Okay, the excess water is given out by the plants, it's given out through the stomata, but now in the night, since the stomata is closed and water is going on coming in, when it's a cold night and the soil is moisture rich, okay, it's properly having good moisture, so the water potential increases and it starts going up the roots. Okay, okay, now what happens is, since there's good amount of water here, the water enters the roots and goes in. Okay, there's already water inside the plant. Okay, there is water inside the plant already. Now what happens is, since water is not able to escape through the stomata, okay, what happened to the stomata? Since it's night, it's closed. Okay, since it's not able to go out, since it's not able to escape out into the environment, what happens is, the water keeps building inside. The water keeps building inside. Where does this water go? Okay, this water is actually exuded out. Okay, let's use the term exude. Exude is to give out. Okay, is exuded out through particular structures called, they're pores basically, called water glands, also called as hydrathodes. Hydrathodes. Okay, hydrathodes. What happens is, since the pressure inside, the water pressure inside the plant is increasing. All the water has to go out somewhere, and the only outlet was actually stomata. Since stomata is closed, it has to find a new way out, and that is through the hydathodes. Okay, and when water is exuded out through the stomata, it's done in gaseous phase. But this thing, through the hydathodes, it actually comes out as water droplets. In fact, if you wake up early in the morning after a particularly cold night and the soil is also well drained, what happens is on the leaf edges, okay, let's draw a leaf. Okay, on the leaf edges, what you see is tiny droplets of water. Okay, tiny droplets of water all balled up. Okay, ready to roll off. Now these things, do not confuse it with dew. Okay, do not confuse it with dew. Dew is actually the condensation of the water vapor that is in the atmosphere. It's not coming from the plant, it's settling on the plant. Okay, there is atmosphere here, and there's water vapor. What happens is the water vapor on cold nights, it condenses and it just settles on any cold surface that is there. It starts settling on top of that and condenses. Okay, now that is dew. Okay, do not confuse dew with guttation. Guttation is coming from inside the plant. Okay, now these droplets are your results of guttation, and they are coming out of hydathodes. Okay, now that is now we've understood what guttation is. Let's try and find out okay, guttation is the loss of water from plants in liquid phase. Let's see which one of these. Water loss in liquid phase. Great. Okay, so D is 3. Let's try and find out if we can apply our previous trick to this. Okay, right off the bat, you can eliminate this and this. Okay, so the contest is between these two. Okay, and you can also see, since these two are already 2, A is 2. You can directly go for it. Okay, let's understand what's A. A is cohesion. Okay, and according to the option, it says that it's mutual attraction among water molecules. Okay, let's understand this. Okay, let's understand this as well. Now, water shows a property of cohesion and adhesion. Cohesion and adhesion. Okay, now what is cohesion and adhesion? Okay, you might have heard the term adhesive. If you've taken your Fevicol bottle and you turn it around, okay, on the wrapper it's written adhesive, strong adhesive. Adhesive means to stick. Okay, adhesion meaning to stick. Now water shows both these properties, and they are both basically the same, that is sticking. It is to what are these molecules sticking is the difference. Okay, cohesion is when suppose, let's say there's one water drop and there's another water drop, and these two water drops stick. Okay, and these two water drops stick, or let's say molecules, it happens at molecular level also. Okay, these two molecules stick. Okay, now that is called cohesion. The attraction and the sticking, okay, of water molecules to each other is called cohesion. Cohesion. Adhesion is the sticking of water molecules to other surfaces, mostly polar, because water itself is polar. Okay, water itself is polar. It has a slight polarity, and because of this polarity, the positive, negative, they attract, and water molecules are able to attach themselves to other polar surfaces. Okay, and that is adhesion. Okay, so let's revise. Okay, let's just go through it again. Cohesion is the attachment of water molecules to other water molecules. Okay, and adhesion is the attachment of water molecules to other polar surfaces, because water itself is a polar molecule. So let's based on this, let's try to match. Okay, cohesion, as we've already seen, is cohesion is mutual attraction among water molecules, meaning they are attracted to each other itself. So that is cohesion. So this is correct. And next, adhesion. What did I say adhesion was? That water molecules are able to stick to other polar surfaces. Which one of it is? Okay, attraction towards polar surfaces. There you go, we've solved it. So B is 4, and that automatically leaves us with C is 1, that is surface tension is the more attraction in liquid phase. So which of these is the correct answer? B4, whichever has B4. Yeah, A is the correct answer. Okay, A is the correct answer and C is 1. C is 1. Yeah, correct. But of course, I'm not going to leave you without explaining what surface tension is. Obviously, I'll be explaining you what surface tension is. Now, let's imagine a drop of water. A drop of water. There are lots of water molecules inside. H2O, H2O, H2O. Okay. Now you have to imagine that this ball of water, this drop of water, has lots of molecules that make up the surface of the drop. Okay, now imagine that this is the drop. The surface of the drop has lots of water molecules in it, and then on the inside that makes up the entire bulk of that drop. Okay, how about I draw it like this? Okay. Okay, so this is the entire drop and this is the surface. Okay, this is the surface and this entire surface is filled with H2O, 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 H2O. Okay. Okay, so this makes up a layer, this surface layer, okay, of just H2O molecules. And then on the inside is also you have H2O molecules making up the entire bulk of the drop. Yeah. Now what happens is, because of the polar nature of water, the entire bulk actually attracts the surface molecules. Okay, so yeah, we've already answered more attraction in liquid phase. Okay, there are more attraction of the molecules of water in the liquid phase. So the outer surface is attracted by the inner bulk, and this forms a small, a layer of water, which is actually used by insects. You must have seen insects actually, striding over water. Water strider sort of insect. Okay. Now this insect actually walks on water, like walks on water in air quotes. Okay, because they do not break the surface tension. The surface tension, what did I say? The bulk of the water attracts, the liquid phase. The bulk of the water attracts the surface, and this attraction starts forming, a thin film of water that is usually not broken. And these insects that walk on top of them do not break the surface tension. Okay, since they do not break the surface tension, they can walk on it because it acts as a layer, it acts as a film, a very thin film at that, but yes, they are able to exploit this particular nature of water. That is our surface tension. And a little extra knowledge that I'd like to give you here is, as I said, since there is attraction from inside, okay, there is attraction from the bulk part of the drop, what happens is, it is this very property of water because of which the raindrops that we have are spherical in nature. Okay, I know we tend to draw raindrops like this. Okay, we tend to draw raindrops like this, but that is not the case. Raindrops are actually spherical in nature. They're actually spherical in nature, and there's a reason. The reason is surface tension. How? Because as I said, the inner bulk starts attracting. It attracts the molecules that are on the outer surface, and this attraction actually, leads to a certain shape that has the lowest surface area. Okay, and it's usually the sphere. It is the sphere that has, when the volumes are equal, it is the sphere that has the lowest surface area. Okay, and it is because of that your raindrops are spherical in nature. Again, just connections. Whenever you learn something, whenever you learn a small piece of information, learn to connect it with other pieces of information. So what happens is, when you are asked a particular question, okay, and okay, so this is the answer, but at the same time you are automatically recalling all the other pieces of information. Okay, so it helps you recall other things faster, it makes you a more knowledgeable person. At the same time, it helps you clear exams, you know, in a much better way, because suppose if you make a mind map of everything that is in biology, while you're answering one question, your mind is already recalling another piece of information that might be useful to answer another question in the very same question paper. So this has happened a lot of times. Okay, so try creating mind maps for yourself. Whatever works for you is best, but this is a general, you know, advice or a trick that I myself use most of the times. Okay. Okay, so we are done with this. This is your correct answer. Okay. So let's move on to the next question. We are in question number 18. Okay, this is pretty much a direct question. If you know the answer, you know the answer. You do not know the answer, you do not. Okay, DNA strands on a, yeah, DNA strands on a gel stained with ethidium bromide. Ethidium bromide is also written sometimes as ETBR. Ethidium bromide, when viewed under radiation, appears as, the answer is bright orange bands. Bright orange bands. So this is something when you suppose if you take up life sciences as your subject, this is one of the experiments that you do. You actually cut up DNA and then you put them on agarose gel electrophoresis, and when they separate according to their sizes, you spray it with ethidium bromide and put the entire assembly under UV light, and you can see bright orange bands of DNA. Okay. Yeah, since the question is asking about DNA strands, the color is bright orange. Okay. There are other things that are used to, there are other chemicals that are used, like ninhydrin is used to stain other biomolecules as well, and the colors are different. But for DNA, when you use ETBR, that is ethidium bromide, the color that you get is bright orange bands. And how ETBR works is, suppose you have your DNA like this, okay, and you have your base pairs. These are your base pairs. Okay, these are your base pairs. What does ETBR do? It intercalates itself. Okay, here the term intercalate. Sir, what is intercalate? You take your fingers and you put them like this. Okay, intercalate. Okay, so they intercalate themselves. These molecules of ETBR intercalate themselves. You know, they basically embed themselves between the nitrogen bases. Okay. So basically, when this happens, when something is put inside, in the central space, okay, there is obvious unwinding of the DNA. Okay, the DNA unwinds a little bit, but anyways, they get embedded themselves. They embed themselves inside, this a DNA strand, and that's how, when you put it under this thing, under UV light, these things start shining, and they shine with a bright orange color. Okay, and since there's unwinding of DNA that is happening, when we used to work in our labs, we were in fact very strictly told to wear gloves and other protective gear, like eyewear and all, because these are said to be carcinogenic or tautogenic. Okay, because there's clearly unwinding of the DNA happening. But again, for full disclosure, when I was trying to go through, for this very same class, when I was trying to go through sources, there are a dearth, there is a little less information, theoretical proof, actual proof, practical proof to show that ETBR is indeed a carcinogen. But yes, when using in lab, it is important to use proper safety equipments. Do not take lab safety lightly. Okay, be very careful when you're handling chemicals. Okay, so that is also done. Okay, coming to botany. The term used for transfer of pollen grains from anthers of one plant to stigma of a different plant, which during pollination brings genetically different types of pollen grains to the stigma, is? I'll tell you the answer directly right now. Okay, and then we'll go through each of these one by one. Okay, The answer is xenogamy. Xenogamy. Okay, let's xenogamy. So since this is the answer, this must be the explanation for xenogamy. Yeah, I'll just explain it again. So there is one tree, there's one plant. Okay, there's one plant, and there is another plant of the same species, obviously. Okay, and then there is a flower here. Okay, and then there is another flower here. They're both genetically different. Yes, they're both genetically different. So let's say the anther, the pollen grains from the anther of this flower. Okay, let's say this is the male and this is the female. Okay. Okay, so the pollen grains come and pollinate a different individual of the same species. They pollinate a different individual of the same species. So this is called xenogamy. And since they are both genetically different, the offspring is also genetically different from both the parents. Okay, because genetic recombination happens and all those things. When, you know, zygote syngamy happens, there is recombination that happens, all these things. Okay, so that is there. Okay, so we've understood what xenogamy is. Let's understand chasmogamy and cleistogamy. Okay, let's understand these two terms. Now, what happens is, chasmogamous flowers is just what we've understood so far. Chasmogamous flowers and cleistogamous flowers. Okay, now chasmogamous flowers are your normal flowers, you know, with exposed anthers, okay, and exposed anthers and stigma. Okay, these are your normal flowers where, you know, cross pollination takes place, that is cross fertilization, what we just understood, xenogamy. Okay, these are your normal flowers, you know, imagine a normal flower, that's your chasmogamous flower. Now, what is a cleistogamous flower? Now, these cleistogamous flowers do not open. Okay, their anthers and their stamen, okay, their gynoecium and andrecium, their gynoecium and andrecium, they are both covered. The stamen and the stigma, they are both covered. So what's happening? A pollen grain that is coming from outside cannot pollinate this flower because they are both covered. Okay, the stigma is covered. Okay, and the anther is also covered, and they are both lying close to each other. They are both lying close to each other. Why is this being done? Why does this plant, why does this flower do not want to get, you know, why does this flower do not want to get cross pollinated? Now, the thing is, in nature, pollination is not always a surety. Okay, it is not 100% sure that cross pollination has to take place. There might not be pollen grains around, there might not be other individuals around, sorry, not pollen grains, there might not be, you know, pollinators around. Okay, there might not be pollinators around, there might not be other individuals around. So what happens? That species, that individual, you know, dies off without having offsprings. So in order to ensure that there is a set of seeds that will carry on, that there will be offsprings, even though they are genetically identical, because they are coming from the same plant, right? They are coming from the same plant and from the same flower. So they are going to be genetically identical. Okay, being genetically identical, having offsprings that are not you know, different from the parents is a downside, is a definite downside. But the bigger downside is not having any offsprings at all. Okay, now that is the bigger downside. In order to avoid that, these cleistogamous flowers are actually closed, and the anthers and this carpel, the stigma, they both lie close to each other, so that definite fertilization takes place and there is an assured seed set. Okay, in order to assure a seed set for the next generation, we have cleistogamous flowers. And obvious differences is the fact that chasmogamous flowers, you know, they have exposed anthers and stigma, whereas cleistogamous flowers, have covered anthers and stigma. Okay, that's one. The next one is, since chasmogamous flowers have to get cross-pollinated, and pollination happens with the help of pollinators, there needs to be, rewards for pollination, like, having nectar or, oviposition. Okay, so chasmogamous flowers produce nectar, or, they might have, places in the flower itself for oviposition, that is for the deposition of eggs, like when I was talking about yucca moth. Okay, okay, the yucca moth, the flower that is there, they give oviposition place for the moth itself. Okay, but for cleistogamous flowers, since they do not need pollinators, since they do not have to get cross pollinated, they do not need to attract any of these pollinators, and because of that they do not have nectar. And also chasmogamous flowers have, vibrant petals, colors, and, scents in order to attract pollinators, whereas cleistogamous flowers do not have any of these needs, because they do not have to attract anybody, so they are fine on their own. Okay, they do not have these vibrant colors or smells or anything. They are just there to make sure that there is an assured seed set for the next generation. Okay, we have understood chasmogamous and cleistogamous. And finally, geitonogamy. Now, geitonogamy is genetically basically autogamy. Okay, it is autogamy. Okay, we haven't discussed autogamy. Autogamy is when, okay, let me just clear the board. Okay, autogamy is when the pollen grains of one flower pollinate the stigma of the same flower. That's autogamy, self-fertilization, right? Now, geitonogamy is genetically autogamy, because what happens is, suppose you have a plant. Okay, this looks like a tree. Okay, now there are going to be many flowers here, right? Let me use a red color in order to, differentiate. Okay, there are going to be many flowers here, right? They're all from the same plant. They are all from the same plant. So each of these guys, each of these flowers are genetically identical to each other. But what is happening in geitonogamy is that the anthers, okay, the anthers of one flower is pollinating the stigma of another flower. So, when you look at it practically, it looks like, it is cross pollination that is happening, but genetically it's basically autogamy itself. Okay, because these two are genetically identical. So, yeah, if you take cleistogamy, the pollination happens within the same flower. Okay, within the same flower, the anther and the stigma are from the same flower. But here, the anther and the stigma are from different flowers, but they are from the same plant. So that is your geitonogamy. Okay, and xenogamy, just running back again, xenogamy is the flowers are from different plants. Okay. Geitonogamy is different flowers but same plant, and autogamy is same flower, same plant. Okay. So we've cleared up on all these concepts. Let's move on. Okay, let's move on to the 10th question. Okay. Okay, which of the following algae produces carrageenan? Okay, which of the following algae produces carrageenan? This again is a direct question. So I'll be explaining what carrageenan is. Carrageenan is basically a hydrocolloid. It's a hydrocolloid. That is, it holds on to water, and this particular substance, carrageenan, actually finds a lot of use in your cuisines, in your industry that produces food, in your food industry. Okay, so when you have Gordon Ramsay coming up and saying, "Oh, good I might, oh, these flies, they're having a party in my mouth," that industry. So texture is a really important part of your food industry. Your sauces have to be of a particular consistency. Your jellies, your ice creams, your ice cream shouldn't be too hard, it shouldn't be too runny, it has to be of a particular consistency. Okay, if you've seen MasterChef, Gordon Ramsay is always picking up the sauces, and he's running it like this. He says that it should coat the back of the spoons, might. It should coat this back of the spoons. Now that thing, the consistency, although it's not used in sauces, it's used in jellies. Okay, it's used in ice creams to correct the consistency. Okay, how much amount of water is being held? Is it runny? Is it thick? Is it smooth? Okay, in order to correct that, we use carrageen. Okay, and the question is, where is this carrageen obtained from? Again, this is a direct question, so there's not much of explanation to do. It's a factual question. You either know the answer or you do not know the answer, and the answer is that it comes from red algae. And as we've already studied from the previous question of this very same lecture, we were talking about chlorophyceae, rhodophyceae, and phaeophyceae. Are you able to connect it? What is red algae? What is red algae? Red algae is rhodophyceae or rhodophyta. So you can see, as I was already talking about, I told you, right, when you're doing a particular question, if you make mind maps while you're doing a particular question, you know a particular answer and you know this concept, and you remember a very different concept, and you are able to use that concept for a different question later on. See this here. That's exactly what has happened in question number 11. We spoke about rhodophyceae, and again we've come here. We're speaking about red algae here. So you see how important making a mind map becomes. Okay, these are all from the same question paper, mind you. Okay, so red algae is rhodophyceae, and they are the ones that produce carrageen. Carrageen is a hydrocolloid. It's used in the food industry to correct for consistency, thickening, and all those things. Okay, so as of today, we've all again completed 10 questions, and with these 10 questions, we've completed a lot of concepts. I hope all these concepts were, understandable. If you have any suggestions to give, do write it down in the comments. We do read our comments. Okay, and with that, I'd like to end the class here. My name is Ram, and it was a genuine pleasure teaching you all. And yeah, the comment section was, if I have to use the millennial way, it was lit. Okay, it was a genuine fun teaching you all. So have fun, guys, and keep learning and keep joining with Easyhai online. Okay, have fun and take care.